Okay, doke, everyone, let's get started. Good afternoon, everyone. It's a great pleasure to welcome you all to the third Department of Genetics, Edith Saunders lectures. So just a couple of housekeeping matters before we get started. Your microphones and cameras are turned off by default on this Zoom webinar. There'll be a Q&A session after Edith's talks, so just submit your questions using the little Q&A box down the bottom, and I'll ask them, and we'll do as best as we can to get through as many questions as possible. The event is being recorded and will be available later via YouTube. So on with the fun. So we introduced this distinguished lecture to remember and honour the pioneering work of Edith Becky Saunders in helping to establish the field of modern genetics, and her amazing contributions to the advancement of women in science at the start of the 20th century here in Cambridge. She was a remarkable scientist. Hailing from Birmingham, she came to Cambridge in 1884 as an undergraduate at Newnham College, where she met and befriended William Bateson's two sisters. She obtained first-class honours in the natural sciences tripos in 1888. Though obviously, as a woman, she was not allowed to get a degree, and she got a certificate of completion. That's a shocking state of affairs, which took till, I think, 1940 to rectify. After her first-class honours, she was appointed as a demonstrator in the Balfour Biological Laboratory for Women at Newnham College, where it was established through a donation by Arthur Balfour to help provide practical facilities for women scientists to do practical work, since they could never get any experience of practicals in laboratories in the university. After that, she had a college lectureship and a role of Director of Studies in Newnham, and there are many tributes to the superb mentoring and teaching that she did. Edith was always an experimental scientist, and after a series of influential breeding experiments with something called the buckler mustard, a type of brassica, I think, she began working with William Bateson around 1895. However, it's very clear that her work was independent and very influential in the acceptance of Mendelism in the early part of the 20th century. And it was, as Bateson always acknowledged, a genuine and extremely productive collaboration between Saunders and Bateson. I could go on to list many of Becky Saunders' achievements, but one example is that she was the driving force behind the formation of the Genetical Society in 1919, initially as its vice president, and then later she became president. We've got extensive information on the life and work of Becky Saunders on the Genetics Department website, so I would encourage you to go and visit that. It was all collated by our former librarian, Christine Alexander. So while Becky was never formally a member of the university or the genetics department, and we're honoured in the genetics department to organise to recognise her contributions with this lecture series. And I'll hand over to the current Arthur Balfour Professor of Genetics and Pro-Vice Chancellor for Research, Anne Ferguson-Smith, to introduce today's lecture.
Thanks, Steve. Hello, everybody. It's great to see so many of you here this evening. This evening we celebrate two Ediths, two remarkable scientists who have really been truly influential in the field of genetics. A very, very warm welcome to Edith Heard, who's joining us today. We really wish you could be here in person, but soon. Edith, I'm going to be brief because I know that Edith will have a lot of great science to tell us about, and you're not here to hear me. But Edith is one of us. She did her Part 2 genetics in 1986 here at Cambridge. And I think that this was a good time in genetics, with lots of really interesting and fun people around, and the field of molecular genetics in particular was really burgeoning. From there, she went on to do a PhD at the Imperial Cancer Research Fund labs in London, where she worked on gene amplification and continued working with, actually did some work on YAC libraries and various other molecular cloning contexts, which actually stood her in good stead when she went on to do postdoctoral research in Paris with Phil Abner. And she was in Paris for many years as a CNRS scientist, then as group leader, and then as director of the Genetics and Developmental Biology unit at the Institut Curie in Paris, which a role that she took on in 2010. In 2012, she was elected as a professor in the Collège de France, where she is chair of epigenetics and cellular memory. 2013, she was elected as a fellow of the Royal Society. And then in 2019, she took on the director-generalship of the European Molecular Biology Laboratories in Heidelberg, and she now has a research lab in Heidelberg. I could go on talking about Edith's accolades and contributions to her wider contributions to the field, but I'd like to come back actually to her science. Over the years, Edith's work has really exquisitely deciphered the process of mammalian X inactivation during development, during evolution, within tissues, within and across generations. But not only has her work really uncovered the mysteries of this really remarkable mammalian process, but her work has also been a paradigm for elucidating new concepts in epigenetics, in functional genomics, and in chromatin organization. I'll stop there actually, because I'm sure she's going to tell us about all these things. Edith, over to you.
Great, thank you so much, Anne. I wasn't sure whether I should appear on the screen or not, so I decided not to, but thank you for the really kind introduction and also for the invitation. I was so thrilled indeed to be invited to give this very prestigious lecture, not only because it's where I discovered genetics and set me up for the rest of my life, basically, the Department of Genetics, but also because it's another Edith, and it turns out she was born exactly 100 years before I was, so it all feels kind of, logical. But also I just want to thank Anne, who is a colleague in the epigenetics field from a long, long time ago, and I feel like in many ways we grew up in the same universe and have seen the whole field evolve into many different ways. So it's a pleasure to be here, and I'm going to now try and share my slides and hopefully tell you a bit about the science indeed that my lab does. So can you tell me if that looks okay, or should I swap?
You need to swap.
Again.
Got your presenter. That's the one. Excellent.
Okay, so it all looks good now, does it? Good, okay, perfect. So actually the image I'm showing you here, I'm going to come back to later on in my talk, but I want you to take note. In this image, you can see several breast cancer cells. In green is the X chromosome. Normally, a female only has 2 X's. You can see there are 3 here. And in red is exist RNA, a key molecule in X inactivation. I'll come back to this later, but you can see the RNA is kind of floating around in the cells. And I apologize for those of you who are Daltonian. I'm sorry. So X inactivation is the process that I've been working on for decades now, and it really is a fascinating phenomenon that evolved, we think, to deal with dosage compensation for the sex chromosomes between males and females in mammals. So it was, in fact, discovered by Mary Lyon, who, by looking at various types of mammals, but mice in particular, realized that heterozygous females for X-linked traits, in particular coat color, tended to have mosaic phenotypes, so patches of fur that were either gray or white, as you can see here, in this particular case of a tabby heterozygote female. And just based on these sorts of observations, as a geneticist, she came up with the hypothesis that during early development in females, one of the two X chromosomes becomes silenced, so that transcriptional activity is reduced to almost nothing for almost all genes, and that this process happens randomly, affecting either the paternal or the maternal X chromosome early on in development. And then this is somehow memorized or propagated through cell divisions, leading to clonal populations of cells expressing either the paternal or the maternal X. And of course, if the female is heterozygous for an X-linked trait, then you see this mosaicism. So her observations back in the 1950s and 60s led to the hypothesis of X inactivation. But the meaning of this process and why it had to evolve is something that I think is still very much out there. It's one of the big questions. We know that the X chromosome is huge compared to its Y homologue in male. So the Y is now just reduced to just over 100 genes. And the X has well over 1,000 genes. And we know that a double dose of more than 1,000 genes is probably lethal. In fact, we know that if you fail to initiate this process of X inactivation early on in development, this leads to early female embryonic lethality. So, but did this process evolve for all X-linked genes? Do all X-linked genes become silenced? Just how global is this phenomenon? And these are all questions that are still very much out there. But back then, back in the 1960s, I think it took quite a lot of insight from Mary Lyon and some of her colleagues who had also observed unusual phenomena associated with the X chromosome that I'll come to in a minute, to come up with this hypothesis. And I just want to spend a couple of slides thinking about the mosaicism that X inactivation leads to. And this is not just in terms of coat color and fur. There are more than 1,000 genes, as I said, on the X, and many of them are very important for housekeeping activities. You know, there are all sorts of housekeeping genes, there are transcription factors, there are enzymes of all sorts. So it's really a chromosome that harbors many different functions. And the mosaicism leads to a great deal of variation within females. And so because the process is set off randomly and stochastically, the proportion of cells that will express either the paternal or maternal X varies. And the way that these cells are then distributed during development can also vary. So in this beautiful example here, I'm showing you from the Nathan's lab from several years ago. They created an X-linked copy of HPRT, which is one of the housekeeping genes on the X chromosome, that was tagged either with a green fluorescent protein or with a red fluorescent protein tomato. And these mice therefore express, when they're female, express either the red version of the HPRT gene or the green version of the HPRT gene, depending on which X chromosome was silenced. And this is just to illustrate how between almost genetically identical siblings, you can see a great deal of variation just in skin fluorescence. This is shining under a UV lamp. This is what you see. And then if you go in and look at different tissues, you can see this extraordinary distribution of red and green cells. And this actually varies between individuals. So what's beautiful about X inactivation is that you see phenotypic variation within the same individual, because some cells are expressing one allele, some are expressing the other, but you also see a great deal of mosaicism between individuals. And in this particular paper, they actually showed some cross-sections of siblings, so cross-sections of the brain. And in this particular mouse, you can see that the left-hand side of the brain seems to be expressing mainly one X chromosome, the red tagged chromosome, and the other half seems to be expressing the other. So, you know, paternal and maternal expression are very, very asymmetric here. But in fact, if you look at other pups in the same litter, you see completely different distributions. So this variation between individuals, this mosaicism, is extremely marked. And it has all sorts of consequences that I thought it would be interesting to think about that were actually raised in this paper, that this diversity actually leads to a lot of different variations, but it's due to an interplay of, as I said earlier, the ratio of founder cells to adult cells, the timing and extent of cell migration during development. And of course, this can actually also be influenced by other loci, autosomal loci as well. And during development, and this is something that we're realizing more and more, some alleles may be counter-selected in specific cell types. So there are many genes on the X chromosome that lead to severe developmental or adult disorders in males. I can just cite a few, hemophilia or Duchenne muscular dystrophy, that have dramatic effects in male individuals because there's only a single X chromosome. In females, one often sees absolutely no symptoms at all. Hemophilia is a very good example of this, and that's because the X chromosome carrying the mutant allele becomes inactivated in half the cells, and then there's a selection for that half of those cells so that only the wild type X chromosome is found to be expressed in 100% of the blood lineage. So there's selection against those cells expressing the mutant allele. And so this kind of variation happens probably at many, many X-linked loci during development and even during in adult lineages. So X inactivation is really a diversity generating mechanism and leads to all sorts of reflections about how different females are between themselves and of course with males. And another layer variation that was discovered some years ago and was initially reported in detail by the Willard lab is the capacity of some genes on the X chromosome to escape X inactivation. So although most genes on the X chromosome become silenced during this process, in fact, an increasing proportion have been found to escape. Up to 10% of genes on the X chromosome on the human X can escape constitutively, in other words, in all tissues. And another 15% of genes, at least, seem to show variable or facultative escape. And this varies between individuals and tends to be more tissue specific. So this leads to an even greater deal of somatic mosaicism and therefore variation between individuals in females. So just to quote Hunt Willard at the time, he made this citation that basically, genetically speaking, if you've met one man, you've met them all. But and we men, he says, hate to say predictable, but women are extremely variable and there's a lot of variation, and it's not Mars or Venus, but every single planet and many more. So this is all very well, but exactly what is the degree to which escape happens, and does it matter? And I just want to say a few words here, and then I'll hopefully come back to it right at the end of my talk. So some of the genes that escape X activation have homologs on the Y, and that's probably the reason why they escape, because indeed you don't need to dosage compensate because males and females are equivalent for X-linked and Y-linked activity. However, many of these escapees do not have homologs on the Y. And so why do these genes escape? Some of it may be accidental. It could be epigenetic instability. Some of it might be purposeful. In other words, it's actually a requirement, maybe because of specificities to do with X inactivation, a requirement for a double dose of some gene products on the X. And in fact, we published a paper a few years ago showing that one of the escapees, UTX, may have a role in the reprogramming of the inactive X during development. And one important factor I want to highlight is that more and more of these escapees are thought to underlie some of the sex-biased diseases that are becoming focused on, including neurological and metabolic disorders, but also autoimmune phenotypes, including, for example, genes such as the toll-like receptor 7, TLR7, which is now known to be very much the genetic basis of lupus, but in a very dosage-sensitive way, which is why more than 90% of lupus patients actually are female, and that's because this gene escapes X inactivation, and it does so rather variably. So this is just to show that actually the degree of escape from X inactivation varies, or is thought to vary, between human and mouse X chromosomes. But in fact, we're finding more and more genes on the mouse X also can escape this process. So just to end on this rather broad topic of sex bias in disease and escape, of course, hormones are often involved in sex biases in disease. And I'm showing you here some of the known sex biases, for example, in cancers, in infectious diseases as well. There's more and more interest in this, of course, with the pandemic as it goes, but also in autoimmune diseases, there's a very strong female bias. And it's turning out to be probably linked to the sex chromosomes. And so it seems that up to 6,000 genes of the 23,000 total in humans are actually differentially expressed in men and women. And this is not just due to hormones, but also to the sex chromosomes, and in particular to some of these genes that manage to escape. And I think one thing that one has to bear in mind about this is that, in fact, most women, when treated, are actually treated as mini men. The medication that we're given is actually based on clinical trials performed on men. This is changing now, obviously, but still many of the drugs, and in fact, a high proportion of drugs that are taken off the market in the past decades have been taken off because of the side effects they have in women. So my one sort of slide about, you know, why women are so precious is that if we're thinking about precision medicine, we should be thinking about treating half of the population in an appropriate way. And there are many genes on the sex chromosomes and on the X in particular, where dosage does matter and variation of X-linked gene expression does matter. And so I would say that we have to move towards understanding how we treat patients based on their sex chromosomes, as well as an awful lot of other factors. So now I'm going to actually talk about some of the work that my lab does to understand how X inactivation happens. And as Anne mentioned, we've been interested over the years in the when, the where, and the how of X inactivation. So this is a very simplistic cartoon that illustrates the big problem, or the big issues, the challenges associated with X inactivation. So unlike most other strategies of sex chromosome dosage compensation in other animals, in mammals, this process of randomly silencing one of the two X chromosomes is quite challenging, because it means that one has to monoallelically express genes in the same nucleoplasm. One has to differentially treat two chromosomes that could, in theory, be genetically identical in the same nucleoplasm. And so how do you do this? And so for many years following Mary Lyon's discovery, there's been a hunt for what is actually underlying the initiation of this process during early female development. And one of the main features of initiation of X inactivation is that it's been mapped down to a region on the X chromosome known as the X inactivation center. And this is where I began my life in X inactivation. It was hunting what actually controls the process of initiation of X inactivation within the X inactivation center. And I was lucky enough that when I arrived in Phil Abner's lab, the Exist non-coding RNA had just been discovered by several labs, including Sahela Rastons, Hunt Willards, and Neil Brockdorf, and Phil was also involved with Andrea Belabio. And that made my life a lot easier, because we actually knew what the molecule was that leads to initiation. And this is this non-coding Exist RNA that coats the chromosome in cis and leads to silencing. But still, that didn't explain to us how X inactivation is triggered in a female specific way when there's more than one X chromosome. And so here I'm wrapping up in one half of the slide several decades of work, but there are still some very interesting open questions about Exist in the X inactivation center. It's a very large locus. In fact, the regulatory landscape of Exist is very big. It's several hundreds of kilobases, and it was in the hunt for how Exist is regulated monoallelically in females during early development that we stumbled across the organization of the X chromosome and of this region in particular into topologically associating domains, which are 3D entities within which sequences tend to interact. And we realized that Exist and its 5' regulators, positive regulators, are organized into one of these topologically associating domains, or TADs, and that an antisense to exist, TSIX, which negatively regulates exist, at least in mice, is located in another TAD. And it could be that the flipping between these two TADs and their activity could help underlie the initiation of exist expression in a sort of stochastic way. But still, there are many open questions, and actually several labs are working on this now, including some of the people who emerged from my own lab. And I would direct you to some very beautiful studies and recent reviews, for example, from Edda Schultz's lab, where she came up with a hypothesis of a symmetrical toggle switching method for, or mechanism for how exist is monoallically regulated. But that's just the beginning. How does then exist trigger the process? And that's what I'm going to be telling you about mainly today. But I wanted to start by saying that once exist has coated the chromosome, it induces a number of changes, both at the chromosome organization level, and this is something that in fact Barr, a cytogeneticist, had noticed even before Mary Lyon came up with her hypothesis. So in 1949, he published a paper showing that in cat neurons, there's what's known as a Barr body of inactive X chromosome, of the inactive X chromosome, which he didn't know was the inactive X at the time, but which seems to be found specifically in female cells, and that's why it is called the Barr body. And this is the heterochromatic entity that is the silent X, the inactive X. And subsequently, using fluorescent microscopy and other techniques, we and others were able to show the organization of the inactive X as a more sort of a spherical entity. And more recently, with chromosome conformation capture technologies, which allow you to capture the three-dimensional structure of chromosomes in the nucleus, we were able to show in collaboration with Job Decker's lab that the inactive X is actually organized into two megadomains, but otherwise is rather structureless, except for a few regions that have these topologically associated domains that I mentioned earlier. And these regions that show these TADs are actually regions that escape X inactivation. So I'll come back to that briefly later. But now I want to just say a word or two about how X inactivation is established, the when and the where. And so for many years, my lab was working on the mouse, trying to understand, how is X inactivation set up? And it turns out that previous work from Tagaki, a Japanese scientist, and also even before that from people like Mary Lyon, had actually stumbled across the fact that X inactivation is initially imprinted. It's only the paternal X chromosome that becomes silenced early on in the mouse. And this imprinted form of X inactivation, it turns out, is due to an exist imprint, which has only recently been unraveled and discovered. And the exist imprint is actually regulated by polycomb. In the maternal lineage, this shuts down exist. So you can't get exist expressed from the maternal X chromosome, but it is expressed from the paternal X. So this imprinted form of X inactivation is actually much more sensible if you think about it, because the choice of which X chromosome has to be silenced is already made. But as I told you earlier, in fact, X inactivation is usually random in mammals. We know that, including in the mouse. And it turns out that this imprinted form of X inactivation, which is set up starting at the two-cell stage and is complete by around the blastocyst stage, in fact, in the inner cell mass of the blastocyst, which are the cells which will form the embryo proper, the epiblast, in the inner cell mass, the paternal X is reactivated, reprogrammed, and then you get the onset of random X inactivation, which can be recapitulated, in fact, in embryonic stem cells. And it was this reprogramming process that, in fact, my lab discovered along with Neil Brockdal's lab several years ago. And it's rather remarkable because basically you set up a silent state of the inactive X through several cell divisions, and then you completely bypass this or override it in the space of just 1 or 2 cell cycles. And then you initiate this random form of X inactivation where either the paternal or the maternal allele of exist will be expressed. And this is the switching process I mentioned earlier that we're still trying to understand. But once exist coats the chromosome, be it paternal or maternal, it will shut it down. And then this will be stably propagated, just as Mary Lyon had predicted in her early studies, and it will be maintained. However, in the germline, the X chromosome will be reactivated so that in oocytes, both X's are in an active state. So I just want to say that after looking at this for many years in the mouse, we decided to then zoom in and look at what happens in other mammals. And here I just want to say that this is where the surprise came, because we believe that imprinted X inactivation might have been perhaps some ancestral way of doing it, because, for example, in marsupials, this is the only way that X inactivation happens. It's always imprinted, but it turns out that exist is not conserved in marsupials. And recent work from James Turner's lab has shown that another molecule, another non-coding RNA known as RSX, could do the job. But it turns out that actually in other eutherian mammals, including humans, rabbits, and more recently in the macaque, X inactivation doesn't seem to be imprinted at all. It actually becomes randomly initiated and with very different dynamics to what I just showed you in the mouse embryo. So although exist is present and is probably doing the job, in fact, exist is biallelically expressed in females. And then once X inactivation begins, it somehow backtracks and you only see one of the two X's coated in, and it's silent. So this is a truly very different way to do this. And in fact, I want to point you to a recent beautiful paper that came out from Michinori Saito's lab, where in fact, Iku Okamoto, who was the postdoc previously in my lab, who did the beautiful work that we published in the humans and in the rabbits. So Michinori and Iku published this paper where they looked in the macaque and they managed to map out the whole X inactivation cycle right through from early development through and into the germline. And they show that indeed the rules are very different to what we had imagined in the mouse. So this is not to say that obviously what we and others have been working on in the mouse is not interesting and important. It actually lays down many of the molecular mechanisms, but the dynamics and the epigenetic choreography that happens seem to be rather different in different mammals. So I would say there is no ideal model. Every mammal has its own mechanism and kinetics for achieving dosage compensation. So now I want to actually zoom in and tell you a bit more about how X inactivation is achieved. How do genes actually get silenced? So although exist was discovered back in the 1990s, the actual molecular mechanisms and the genetic and epigenetic factors that feed into this process had remained mysterious for many years. And this, in some ways, has been, the holy grail for people working on X inactivation like me is, okay, we know many of the actors, we know the choreography, but how does it actually work? How do you shut down one allele of every gene almost on the X chromosome and keep the other allele on? And so we know that exist is essential because knockouts of exist have been shown to be absolutely necessary for triggering X inactivation. We also know that if you take an exist transgene and put it onto an autosome, for example, that is sufficient, at least in multi-copies, or if you induce it with a DOX-inducible promoter, that is sufficient to induce silencing in cis. So we know exist is essential, but all of the other factors and how exist does it have remained unknown. So what are the transacting factors? What are the chromatin modifiers? And we do know that there are a number of features of the inactive X at the chromatin level that come in and they're important for its silencing, including the loss of some of the active marks, such as H3K4 trimethyl. So several histone modifications do change, but also DNA methylation comes in as rather a late mark, and it marks the 5' ends of X-linked genes. So we know that these changes happen, but what are the modifiers and how do they work? And also, to what extent are these modifiers then responsible for the memory of the inactive X? And we know that you can take away exist in an adult cell, and genes don't just come back on completely. So it does seem that there's an epigenetic locking system that comes into play. But if I go back to exist, it's a 17,000, so sorry, not 1,700, it's even bigger, 17,000 nucleotide long non-coding RNA. It's retained in the nucleus. It coats the chromosome in cis and it triggers silencing. It triggers these chromatin and conformational changes that I mentioned earlier. And in fact, a few years ago, Mitch Gutman and Engreitz actually published a beautiful paper showing that in fact exist RNA coats the chromosome and hits it wherever the exist gene is associated in 3D space with the chromosome. So the first places where exist actually coats the chromosome are actually the regions that interact with the exist locus, but very rapidly, within the space of just a few hours, exist then coats the whole chromosome and very rapidly triggers its silencing within the space of just a few hours for some genes. So exist RNA exploits 3D architecture, but that still doesn't tell us how silencing works. And then the other feature I wanted to mention, because I did talk about it earlier, is that some genes somehow avoid this whole process, or some genes get silenced and then become reactivated. So there are many genes on the X, as I told you earlier, that can somehow escape or override this process. Most genes on the X do remain very silent in somatic cells. The rate of reactivation is less than 1 in 10 to the 9, which is less than the spontaneous mutation rate. So global stability and yet local instability. And that's something we're very interested in and trying to work out the mechanisms for too. Is it something about the chromatin state of these genes? Is it something about avoiding exist function? And is it something about the 3D organization of the X chromosome? And I mentioned to you that regions that somehow are organized in these TADs on the inactive X, there are very few of them, but they tend to be those regions that escape X inactivation. So these are ongoing projects in the lab. And this is just to show you an illustration of what I mean by the 3D organization of the X, with most of the inactive X not being very structured, but a few regions, these escapees, that seem to be located together and loop out, in fact, from the inactive X chromosome. So I want to zoom in now on some of the molecular mechanisms. And some years ago, when Edith Schultz, who I mentioned earlier, was in my lab as a postdoc, she set up a system together with Christelle Picard, who's my lab engineer, set up an embryonic stem cell line that was very useful in dissecting the molecular mechanisms of X inactivation. Now, normally in a female ES cell, both X chromosomes are active, and only when you induce differentiation do you see silencing of one of the two X chromosomes begin, at least in the mouse, in mouse ES cells. Human ES cells are a different matter, but in mouse ES cells, undifferentiated cells, both X chromosomes are active until you differentiate, and then one of them becomes silenced. And so Edith actually used this system to prove that, in fact, having two active X chromosomes slightly blocks or delays differentiation. You need to inactivate in order to release from pluripotency. But what we managed to create to test that hypothesis actually was an exist-inducible X chromosome that, in fact, Anton Wutz and Rudolf Jenich had created an allele of several years ago. And we made this female ES cell line where we could switch on exist at will using tetracycline or this DOX-inducible system, which meant that we could actually induce X inactivation in undifferentiated ES cells. So we could uncouple it from differentiation, which is an intrinsically quite heterogeneous process. So we could therefore keep the cell state constant, but we could switch off one of the two X chromosomes. And what Edith showed in her paper actually back in 2014 was that, in fact, the gene silencing kinetics that happen are very similar to what we see during normal embryogenesis. And this is work that we did at the single cell level in the lab. So in fact, gene silencing events are very similar, and most of the chromatin events, including the recruitment of polycomb factors that lead to histone modifications, were also recapitulated. So this is a beautiful system for dissecting the precise molecular mechanisms and kinetics of X inactivation. And this is what another couple of very talented postdocs in the lab went on to do. They created an epigenetic roadmap of X inactivation. They took this cell line, they switched on exist, and then they looked after every few hours, 4 hours, 8 hours, 12 hours, 24 hours, using both native chromosome and immunoprecipitation and sequencing, but also RNA sequencing to look at transcription. And we did an allele-specific analysis, and I forgot to tell you perhaps the most important thing about this cell line is that it contains two highly polymorphic X chromosomes. So we can distinguish them at the sequence level through the single nucleotide polymorphisms they have. And we know because the tetracycline, the DOX-inducible promoter is only on one of these two X chromosomes, the black 6 X chromosome, we know which chromosome will be silenced. So that way we can distinguish from the castaneous chromosome, which is the other chromosome and which remains active, we can distinguish what happens both at the genic level, transcription, but also at the chromatin level, chromosome-wide. And using this, Jan Zilic and Aurélie Bouzard, performing allele-specific analysis genome-wide, they were able to show, and then on the left here, I'm just showing you how they did the analysis. I'm not going to go into the details here, but by looking at the D-scores and the allelic ratios of black 6 reads from the future inactive X chromosome and castaneous reads on the always active X chromosome, they could plot the reads from the inactive X to the reads from the inactive X and the active X, and that D-score is what we plot to see how much a gene on the black 6 chromosome is being affected after exist is induced. And by doing native ChIP-seq, we were able to show that indeed some marks are very rapidly changed on the X chromosome, and that includes some of the histone acetylation marks, which we knew are associated with activity and become very rapidly depleted on the X chromosome as it becomes silenced. And here you can see the black 6 and cast reads, black 6 in red, cast in green. And we can see that even by 4 to 8 hours, we're starting to lose reads on the inactive or exist-coated X chromosome. And so what they did was look at many different histone modifications and work out that indeed there seems to be an order of events, and loss of H3K27 acetylation, which is one of the marks associated with activity at gene promoters and enhancers, is one of the fastest marks to be lost. And furthermore, they went on to show that one of the histone deacetylases, which is known to be which remove this mark during development, is actually responsible for this on the X chromosome. So HDAC3 is the only histone deacetylase that seems to affect the loss of histone acetylation on the exist-coated chromosome. But when we looked at chromosome-wide effects, and this is now showing you the whole X chromosome and the allelic ratios plotted here, so you can see that genes that are expressed from the active X are shown in red, and when they're expressed from the inactive X, they're shown in blue, but so mainly red, so active X. So what we could see is that both at the RNA sequencing level and at the chromatin immunoprecipitation level, looking at HDAC3 in H3K27 acetylation, in HDAC3 wild type cells, you can see that the loss of the histone acetylation happens very rapidly between 4 and 8 hours, just as gene silencing does. But then in the HDAC3 mutants, you can see that most genes, but not all, do not seem to lose histone acetylation. And it turns out that indeed there's a big subset of genes that seem to remain acetylated. And although at later time points we find that this acetylation is still lost and these genes still get silenced, there clearly is an effect of HDAC3. So I'm going to just summarize here the different kinetics that we found in this paper. I'm not going to go through all of them. You're welcome to ask questions or to look it up. But basically what we found was that very rapidly after exist coating, within 4 to 8 hours, we lose histone acetylation at promoters and enhancers. But we find that some of the other histone marks are much more slowly lost, including H3K4 trimethylation at promoters and H3K4 monomethylation at enhancers. Furthermore, we showed that polycomb accumulates on the exist-coated chromosome actually much more sharply than previously thought, much more rapidly. But in order to spread into genes, these polycomb marks, which are traditionally thought to be involved in the maintenance of X inactivation, you need to have silenced genes in order for polycomb to spread into the genic regions. So the conclusion of this study was that exist RNA does change chromatin, and it as well as triggering gene silencing, and this does involve some of the chromatin modifiers such as HDAC3, but not only. So that then brings me to the next series of approaches that we used. And in fact, many labs were hunting for the partners of exist RNA. So here I'm just summing up several papers that were published in the space of just 1 or 2 years, where finally, after more than a decade or two of research, people finally started to find out what are the partners, the protein partners of exist RNA that could mediate its silencing mechanisms. And some of these partners are shown here. I'm not going to go through all of them, but exist does bring in several factors, particularly via a region of exist that we previously knew was essential for exon activation, and that was a region known as the A-repeat region of exist, which is the most highly conserved part of the RNA. And some of the factors that were specifically brought down by this A-repeat region of exist included this factor known as spen. And spen was an unknown player in exon activation. There was no previous evidence that spen was involved in exon activation, even though it's a protein that is known to bind RNA and previously been shown to be involved in transcriptional regulation, for example, in the context of hormone responsive nuclear receptor pathways. But people hadn't looked in the context of maternal zygotic knockouts to see if there was an effect in female embryos, which we then went on to do. But spen, I just want to say, is a huge protein. It actually has several different domains that are of interest. It has several RNA binding domains, which is how exist recruits it. But it also has several domains that are involved in signaling and also in chromatin-associated factors, including this Spock domain that you can see at the end here. So in fact, my lab decided to try and dive in to find out how important spen was in exon activation. And this is where François Dossin, who was a very, very talented student in my lab, who's now moved on and he's doing a postdoc at the Pasteur Institute. But what François was able to create was using this beautiful female ES cell line with an inducible exist promoter. He was able to knock in the machinery that would allow us to degrade the spen protein at will using what's known as a degron system, the auxin degron system. So auxin is actually a natural plant hormone which has been modified now to be used with a lot of power to degrade endogenous proteins in different systems. And so by knocking in the AID tag that allows it to be targeted when auxin is added to these cells, so we knocked in to both endogenous spen alleles, and spen is a gene that is autosomal. And so when we knocked in the AID and tag, as well as a halo tag, so we could visualize either halo or GFP, we made different cell lines. We could then actually explore what happens when we degrade spen by adding auxin. And this is shown here in the Western blot. You can see that basically when we add auxin, within an hour of auxin addition, we lose endogenous spen almost completely. But if you wash out auxin, you bring spen back. So it's a beautiful system for reversible depletion, acute depletion of a protein. And so that's what François did. And he could then ask the question, well, what happens to exon activation? If you switch on exist by adding dox, and you take away you take away spen by adding auxin, what happens? And what François was able to show, so here you can see after just 24 hours of dox induction without adding auxin, you can see that most genes on the X chromosome become monoallelically expressed only from the active X. But then if you induce dox, so induce exist, but add auxin at the same time, you pretty much abolish gene silencing. And so this was validated using various other techniques, both qRT-PCR, pyrosequencing, and RNA fish. And we found that indeed most genes on the X chromosome are not silenced in the spen degron when you add auxin. And this is rather different to the HDAC3 knockout, where many genes were still silenced. And we also looked, and I'm not going to show you here for the sake of time, we also looked at the knockout of spen using maternal zygotic deletion of spen. And we found that indeed, by the blastocyst stage, the knockout of spen phenocopies the knockout of exist. So you really don't see gene silencing chromosome-wide when spen is depleted. So how does spen do this? And here I'm going to whiz through a few slides to basically tell you that François went in and dissected which bit of spen is responsible for this gene silencing effect. So what he was able to show, first of all, is that the RNA binding domains of spen are important to be recruited to exist. But the only region of spen that seems to be important for gene silencing initiation is this Spock domain at the C-terminal domain. And basically, when you knock out endogenous Spock, you can no longer see gene silencing. But Francois went on to show that if you artificially recruit the Spock domain to exist, using a system which is a BGL system, whereby we knocked in a series of stem loops into the exist RNA, so we could both visualize it using GFP, and I'm not showing you that for the sake of time, but you can visualize exist, and you can use the BGL protein to tether whatever else you want. It can be GFP or it can be Spock, and that's what Francois did. And what he showed that if you induce exist with this tag, and basically do it in a background where you can deplete endogenous spen, but you bring back the Spock domain, you rescue the loss of endogenous spen. So this was remarkable. We didn't rescue silencing of all genes, but of many genes on the X chromosome. So it showed us that Spock, in this very artificial system, just Spock brought in by exist RNA, is able somehow to induce gene silencing chromosome-wide. And so what's special about the Spock domain? So what Francois did was go in and pull down the different protein complexes that might bind to Spock in embryonic stem cells. And here you can see a summary of this. And what we found was we had some of the known actors. So for example, the NCOR SMART complex, including X-chromosome, which we had independently found to be involved in gene silencing. So NCOR SMART is brought down by the Spock domain. But it also brings down the NERD complex, including CHD4, and it also brings down the RNA methylation M6A machinery, which had independently been found during in these screens that I mentioned to you, where several factors had been picked up to bind to exist RNA in the A repeat. That included some of these M6A machinery factors, and in fact, we find that they bind to the Spock domain of spen. It also brought down factors included in the SAL4 complex, also in RNA splicing and localization complexes, and last but not least, in transcription. And this was a big surprise to us. Basically, the Spock domain interacts with RNA pol 2 and with other factors involved in transcription. So the conclusion of this was that the Spock domain of spen probably brings in a whole panoply of different types of protein complexes to the X chromosome. And what Francois then went on to do, actually he did this in parallel, was to find out where does spen go on the X chromosome. And this then led us to a clue of how spen might be working through the Spock domain. And so to look at where spen goes when exist coats the chromosome, he performed cut and run, which is, I'm not going to have time to tell you exactly how it works, but it's a nice technique that allows you to follow where a particular protein factor or even histone modification is found genome-wide. And using this analysis, which gives you allele-specific resolution, what Francois found was that before you treat cells with DOX, so before you induce exist, spen doesn't seem to bind very much on the X chromosome. It binds autosomes and the X chromosome equally, but as soon as you induce exist, it becomes highly enriched on the X chromosome. As you can see here on the left, within 4 hours, you can see it peak. But then interestingly, it goes down. Even though exist continues to coat the chromosome, in fact, spen becomes released or detached from the chromatin of the X chromosome, even though it still seems to be exist bound. And so when we zoom in and see, well, where exactly does spen go? It turns out that spen binds, well, to the exist gene, which is as would be expected, but elsewhere on the X chromosome, it only binds to active promoters and enhancers. So in fact, spen seems to be targeted via exist to the chromosome, but then spen will only be tethered to active promoters and enhancers. Silent genes on the X, because there are some genes that aren't even expressed on the active X in the ES cells, are not bound by spen. And furthermore, genes that are become inactivated by exist are initially tethered by spen at their promoters enhancers, but then as they get silenced, spen goes away. So we believe that spen is acting via a sort of hit and run mechanism. Exist helps to bring it in, it becomes tethered. And in fact, we've been working on this with another lab at EMBL now, the Muller lab. What we found is that via the Spock domain of spen, which we knew associates with Pol2, I told you that earlier, probably Spock interacts with the C-terminal domain of RNA Pol2, and that could be the mechanism by which it's tethered. So phosphorylated CTD of Pol2 brings in Spock, and therefore spen, and that is what tethers it to chromatin. And Spock, in addition, brings in a whole load of other factors. So we think that, in summary, spen is a key regulator of gene silencing during X inactivation. It's recruited by exist. It's tethered to chromatin by the Spock domain via RNA Pol2. It could be that this tethering also interferes with transcription. That's something we're testing right now. But what's more important is that spen seems to act as a platform for many different chromatin modifiers and chromatin remodeling complexes, including NCOR and NERD. And we think that different genes along the chromosome may be sensitive to different components of this spen platform. And this is ongoing work in the lab. I hope I can tell you more about this, or you will see more about this published in the coming year. I just want to end, though, by saying a few words about escape. I realize that I'm running out of time, so I'm not going to be able to tell you that much more. But I wanted to highlight that in this study, when Francois looked at how a spen affects X inactivation, he looked not only in ES cells, he also differentiated the ES cells into neural progenitor cells. And here he could then deplete spen after differentiation to see what happens to the already inactive X chromosome. So in these neural progenitor cells, spen is already has already done its job, genes are silenced, chromatin modifiers can come in, promoters of X-linked genes are mainly methylated. And so what Francois did was deplete spen acutely using the auxin-degron system in these cells and look to see what happens. And what he found was that actually most cells are completely unaffected. So this suggests that most of the genes that are completely silenced do not become reactivated when you deplete spen. And this goes along with the dogma in the field, which is that you only need exist and its initiation partners early on. But what he did find was that there was a subset of genes that you can see in this little hiccup here in this cumulative fraction. And it turns out that the subset of genes that do become expressed differently are these escapees. So it turns out that the genes that escape variably, that sometimes get silenced and sometimes they can escape, these genes actually become overexpressed. So it seems that exist-mediated spen recruitment still does have an effect, even in these differentiated cells, to dampen the level of expression of these escapees. So they're already expressed, but they become more expressed when you dampen, when you release spen dampening. So does this matter? And in the last part of my talk, I was going to tell you that we think it does, by telling you something about what happens in adult mammary stem cells. For the sake of time, I'm not going to tell you this part of the story. I apologize. It's a very nice collaboration with the groups of Raphaël Margaron and Christophe Genestier and Emmanuel Charaf-Joffre in Marseille, where they had previously made observations that if you knock out or knock down exist in adult mammary stem cells, you see an effect on differentiation of these stem cells. They become blocked and they tend to not show appropriate differentiation into the luminal pathway. And so this links up to previous work that other groups had shown, and this is the first slide I showed you in my presentation, where in breast cancer, it's been shown that in fact exist seems to mislocalize on the X chromosome. It doesn't seem to coat the chromosome as stringently. And in fact, even Barr had noted that in cancer, the Barr body seems to disappear. And that's shown here in DAPI. This is a normal cell up here where you can see the Barr body, but in this cancer cell, you no longer see the Barr body. And so there was this idea that maybe the inactive X becomes reactivated or its chromatin becomes decondensed. And this then linked to this loss of exist or tight exist coating. And so what was done in this paper that was conducted by an extremely talented postdoc, Leah, who worked between Raphael's lab, Christoph's lab, my lab. She went in and looked in a mammary stem cell scenario to see what happens if you deplete exist. And to cut a long story short, she showed that in fact what happens is that you affect SKPs, and some of these SKPs include some key factors such as the mediator gene, mediator 14, which is involved in genome-wide enhancer regulation and remodeling. And this disruption of exist and its partners in a cancer context could have genome-wide consequences. So just a little bit of dosage deficiency can lead to very big effects. And that's why we think that exist and its partners, including spen and polycomb, could be very important in adult stem cell scenarios. And this brings me back to the very first few slides of my talk, where I was talking about facultative SKPs. And we think that these facultative SKPs are very sensitive to exist and its partners, and their misregulation could lead to diseases, in some cases to diseases such as cancer, and in other cases to autoimmune diseases. So I'm sorry to have jumped through the last part. This paper is under revision. It's on Cell Sneak Peek if you'd like to look at it. And this is just a summary of what I've told you. I hope I've managed to convince you that 30 years on, exist at least is starting to release some of its mysteries. But more than 60 years on, we still don't understand how X inactivation really works, but we're starting to get a handle on the molecular actors. And we think that exist and spen, being a master regulator, are bringing in a whole panoply of different chromatin modifiers that lead to gene silencing across the X chromosome. And in some cases, some genes do manage to evade this silencing to some extent. And this can have consequences in tissue homeostasis, stem cell differentiation, and in some of the sex bias disorders that I mentioned when genes can escape and dosage goes wrong. So I want to thank all of the people in my lab, past and present, a very brave lab, many of whom moved with me from Paris to Heidelberg, which was a bit of a culture shock. But we're still going strong and reproducing as well in all sorts of different ways, both productively, scientifically, but also it's a wonderful lab to have around me. And I want to thank my collaborators when I was at the Curie Institute, now that I'm at EMBL, and many other collaborators around the world, and of course my funders. And thanks for your attention.
Thank you so much, Edith. That was a fantastic tour de force along X inactivation with some really nice recent stuff coming through.